Good morning, everyone. Welcome to you all in my presentation. I'm Shohel Rana, PhD student at the University of Queensland. Professor Mark Cooper is my principal advisor. Professor Christian Beveridge, Dr. Owen Powell are my co-advisors. Today, I will be talking about my PhD project entitled Arabidopsis thaliana as a model to investigate accelerated selection response for complex traits in crop breeding. In plant breeding, identifying the best performing individual plants or group of plants for a particular trait in subsequent generation is very important, but how those selected individuals perform in subsequent generations is a matter of concern. Selection response, whether predicted or realized, is a factor of trait heritability and selection differential. In plant breeding programs, it's evident that the predicted response often differs from realized response in the field. This is because the predicted response is estimated using genomic prediction models, which often uses simplified assumptions about how genes contribute to the traits, while the realized response reflects how those genes behave in plants in the field. When the trait is quantitative, like branching in crop plants, its genetic architecture is controlled by a network of genes and physiological pathways, which make it harder for predicting its performance. This is due to the non-additive genetic variation underlying the gene networks, genotype-environment interaction effect, low heritability of the traits, and experimental error that increase the difficulty to get higher prediction accuracy. How do I fit in this scenario? I will experimentally investigate the response to selection by comparing predicted response using genomic prediction models with the realized response observed across generation and selection cycles. A key focus of this work is to understand how gene networks governing complex traits influence this discrepancy, recognizing that traits are controlled by interactions among multiple genes and environmental factors. By incorporating prior knowledge of gene network along with selection pressure, this study seeks to improve prediction accuracy and reduce the gap between expected and observed response. Ultimately, the goal is to enhance the efficiency and reliability of selection by angling genetic model more closely with the biological mechanism underlying trait expression. To do the experimental investigation of selection response for the complex trait, I picked Arabidopsis thaliana as a model plant. A question might strike in mind: why breeding in Arabidopsis thaliana when there is no economic significance of this plant? Arabidopsis have the small genome where function of known genes are well documented. More importantly, life cycle of this plant is shorter, which is ideal for achieving several breeding cycles quickly. And the experimental investigations of this model plant may help to set the principle to approach prediction of selection response for complex traits like branching, which is a very crucial trait in crops. In the experiment, the traits of interest are days to anthesis, that is the flowering time, and rosette branch number. I also had shown days to bolt in the top left corner, which is the first step when Arabidopsis plant shift from vegetative to reproductive phase. The bolting time and flowering time is tightly correlated. And we found the counting number of rosette branches and recording flowering time is comparatively easy. However, the underlying genetic mechanism controlling these traits are highly complex. To test whether the gap between predicted response and realized response increased or decreased for a complex trait and what biological reasons are causing this difference, we have formulated these four objectives. To start with, I will assess the power of predictive selection over breeding cycles and then move on to quantifying the influence of changes in allele frequency across breeding cycles. We will also test the genotype environment interaction associated with light intensity and quantify its effect on prediction model. And finally, we will be in a position to use the G2P models to predict the genetic effects underlying response to selection. To achieve those objectives, we have begun with a population development. For the parental selection, 68 accessions from 1000+1 Arabidopsis diversity panel were evaluated. Then we have selected two parents, H1, 76563 as high branching parent and L1, that is 76482 as low branching parent, which were different for branch number but had almost similar flowering time close to col zero, which is reference accessions. Our thought was that we could remove the confounding effect of flowering time and focus on true genetic difference for branching alone. Now the question is, can we expose the genetic variation for these traits, what we see in this natural population of Arabidopsis? Can we identify the genomic regions that contribute to the phenotypic difference for these traits? Does two trait of interest response to selection as a direct single target? To explore the genetic variation, we did the crossing between selected accessions, which gives us the F1, which is the first filial generation, and selfed them to F2 generation. At this F2 generation, we started two separate breeding programs. Breeding program 1 that have 362 lines and breeding program 2 which includes 450 lines. Here, the aim is to test one of the scientific questions. If we repeat the same selection strategies for the complex trait from the same F2 source, would we end up getting similar estimate of genetic effects? Does the same genetic region respond to our selection? This repeatability of the selection response process within this biparental cross can give us a foundation for understanding the potential accuracy of genomic prediction across breeding programs and cycles. This figure showing the experimental plan I'm working on. The biparental cross was advanced till F5 generation by selfing following the single seed descent method. F5 generation is the traditional mapping population for my study, which will genotype for prediction works. We did some preliminary works in F2 generation to find out the best way to phenotype our traits of interest. Then we evaluated my traits of interest at F2-3 generation and F5 generation. Two breeding programs were evaluated separately at F2-3 generation. Based on the F2 phenotyping result, we started a divergent selection. I will be talking about this divergent selection in later slides. And initially, I had a plan to go till breeding cycle 2, but due to space and times constraint, I will be able to complete breeding cycle 1 during my PhD tenure. When I say breeding cycle, it comprises of three components, that is evaluation, selection, and crossing. Likewise, in my experiment, we evaluated the 1000+ Arabidopsis diversity panel, then selected two parents, and then did the crossing. And the progeny from this crossing will form the base evaluating material for the next cycle. In the overall plan, this part I am calling as cycle 0, and this part will be cycle 1 of my experiment, which will allow me to test our ability to predict from one cycle to another. As my plant is being grown in the growth room, where the plants were placed in multiple racks as shown in the image, each rack had upper lower shelves and multiple trays in each shelf. We analyzed my phenotyping data using the mixed model framework, which allows us to adjust phenotypic values for each of the genotypes for the shelf position, tray within shelf, pots within a tray, and extract the blobs, which is considered as the genetic merit of a genotype. When we did the phenotyping at F2 generation to see whether we could confidently select contrasting individuals for branching and days to anthesis. If that's possible, we could drive multiple cycles of selection in less time. However, the heritability we obtained from primary rosette branch number was very low, that was 0.18. And for days to anthesis, it was 0.46. In addition, we also observed a weak negative correlation between days to anthesis and number of primary rosette branches. As the heritability was low in F2 generation, we decided to replicate each of the line in F2-3. In breeding programs, it's important to know how many replication is good enough to get reliable heritability estimate. So our confidence to select progenies goes higher. To test that, we took 50 F2-3 lines from breeding program 1 and evaluated with 5 replications for a range of traits. And we observed that with 3 replications, we could achieve a heritability value of 0.5 for branching trait, which did give confidence to perform selection. What happens here, when we replicated them, it helps us to average out environmental variation. When we estimate the blob to select extreme individuals, it's an average effect of 3 observations if it replicates 3 times. And we call it as family mean heritability. Here's the data from all the F2-3 line grown in 3 replications from both the breeding programs. And we found the heritability value for rosette branch number was 0.46. This improved heritability gives us comparatively more confidence to select progenies for branching and flowering time. The negative relationship between days to anthesis and number of primary rosette branches grew stronger in F2-3 generation compared to F2. And other trait correlations were higher than that was observed in F2. When we replicated them, it's averaged out the environmental variation. In the F2-5 generation, the heritability value for both of the trait improved compared to F2-3. And what surprised us is the huge change in relationship between flowering time and number of rosette branches compared to what was observed in F2 and F2-3 generation. The relationship became more quadratic than linear in F5 generation. Here I plotted the relationship between flowering time and number of rosette branches in same scale across generation from both the breeding programs combined. What is clear, how segregation over generation shaped the trait relationship. For instance, there was 1 or 2 individuals that flowered before 30 days in F2, 10 to 15 individuals in F2-3, but nearly 100 in F2-5. If we see F2 or F2-3 generation, any plant that flowered earlier are the one that had higher branch number. However, when flowering became too early, the number of branches was lower, which makes sense biologically. We also did the genotype environment study to see whether the difference in growth environment results in different phenotypes. We evaluated all the individuals from breeding program 1 and 2 in 2 environments. In the highlight room, there was 150 micromole per meter square per second. In the low light room, it was 70 micromole per meter square per second. Here, our aim was to test, does the difference in light intensities impact the sugar pathways in branching network? Can it trigger more rosette branch as sugar is a positive regulator of branching? Though we observed the plants grown under 75 micromoles light on an average had delayed 7 days by flowering by 7 days and produced 0.8 less branches. We didn't observe any significant G-Y interaction for both the traits that we are interested in. This is the result from my G-by analysis for days to anthesis. Here, P-geno representing the variance component for flowering and environment, and P-geno representing the G-by interaction, which is very low. And in case of rosette branch number, the environment and genotype interaction variance component is literally 0. In a way, it's an important result as the variation in light is not going to change the phenotype or genotype ranking under different environments. Thus, we could confident about our phenotyping outcomes. Now the divergence selection. In previous slides, I showed how we progressed to F5 generation. Now I am transitioning to how divergence selection of the pools were made. From each breeding program, we created 5 separate pools to represent different types of selection. 2 pools were selected for contrasting flowering time, that is fast and slow flowering, and 2 pools were selected contrasting for branch number, that is high branching and low branching. We also kept 1 pool as a random group without any selection, which acts as baseline to compare how selection changes plant traits. 50 individuals were selected for each of the pools. For creating the flowering pools, the BLUP estimates from the phenotypic data were ranked. Then top 50 ranked individual formed the first flowering and bottom 50 formed the slow flowering pool, as shown in the graph, in the x-axis showing the days to anthesis, that is flowering time, and y-axis showing number of rosette branch number. Here, orange dot, what we have selected as the first flowering individuals, and the blue dots representing the slow flowering individuals. In case of branching pool selection, as we found a negative relationship between days to bolting and number of rosette branch. So to ensure true genetic difference for branching is accounted, we adjusted the flowering time to deduct confounding effect before assigning individuals to high or low branching pools. In the x-axis, we have the days to bolting, and in the y-axis, it's number of rosette branches. Here's the orange dot represents the high branching groups and blue dots represent low branching groups. After ranking blobs, we found most of the high branching individuals bolted within 35 days. And to ensure the desired number as well as to select individual with almost similar flowering time, we restrict our selection for low branching pool before 35 days as well. this slide showing the distribution for flowering time. Here in the orange curve represent the distribution of selected first flowering individual, and blue curve showed the how individual, and the blue curve distribution is the selected slow flowering individuals, and the gray curve show how individuals from whole population was distributed. There were 10 difference between fast and slow, 10 days difference between fast and slow flowering pool, both in F23 and F5 generation. When you advance then F25 by selfing, the difference exists there. But in branching pools, here it represents, blue represents the low branching and the orange represents the high branching pool. In F23, we observed 2.5 branches with difference of 2.5 branches between high and low, which diminished to half a branch in F5 generation. For the biparental cross, after looking at what happened for trait relationship in F5 generations, the way we selected for branching pools at F23 by adjusting for flowering time may not be best way to select. Still, one of our main question is the difference we saw in F23, F5 is additive? Would that genetic variance going to transfer to the cycle 1 progeny or not? After evaluating our cycle 1 progeny, we would be in a position to answer it. So after the selection, I did a pairwise crossing for each of the pool. By intercrossing the selected pools, we allowed them to recombine and segregate even further to release the variation. Per breeding program, it was 125 crosses in total, and 2 breeding programs put together, it was 25 crosses. To obtain enough seeds for further evaluation for a individual cross, I had to make crosses more than 5 birds. So the total number of crosses I made is much more than what I am showing here. From each pairwise crosses from both programs, we decided to advance 10 progenies from each of the cross to keep an equal representation from all the crosses. Now I am preparing for evaluation of the cycle 1 progeny. After evaluating this progeny, we will be estimating the realized response to selection and compare with the predicted response. And finally, the prediction part. To achieve the final objectives of my research work, for the genomic prediction, we already have the phenotypic data, and we are preparing for genotyping of the F5 population. We will build our model at F5 dataset and traditional cross-validation here. And we will try to predict the progeny performance at cycle 1. We will try to build model in cycle 0 and try to predict progeny performance at cycle 1. If that doesn't work, we will move on to build model based on cycle 1 progeny dataset as well. Apart from this, we will have lot more options to predict. From the cycle 0 F5, we will try to predict the 2 breeding programs separately, and also the selected pools. After finishing the progeny evaluation, we will estimate the realized response and compare with the predicted response and see how close they are. So the key takeaway from my research work till now is to get better prediction accuracy for complex trait. It is important to consider nonlinear multi-trait relationship that we saw in F5 generation while building the model. We also realized straight counting of rosette branch number may not be equivalent to precision phenotyping. And to predict rosette branch number with higher accuracy, it's very important to include flowering gene network along with the branching network. That's all about my presentation. I am grateful to ARCCOE for the funding. And I'm also grateful to my supervisor, co-supervisors, and my lab mates. Thank you, everyone.